Hello everyone. Today's topic is autonomic nervous system. The autonomic nervous system is the part of the nervous system that controls most visceral functions of the body. This system helps to control arterial pressure, gastrointestinal motility, secretion, urinary bladder emptying, sweating, body temperature, and many other activities. The autonomic nervous system has three branches: the sympathetic nervous system, the parasympathetic nervous system, and the enteric nervous system. Some textbooks don't include the enteric nervous system as part of this system. The sympathetic nervous system activates the work of most organs except for digestive process. It is often considered the fight or flight system, while the parasympathetic nervous system, it is an antagonist of the sympathetic, is often considered the rest and digest or feed and breathe system. In many cases, both of these systems have opposite actions, where one system activates a physiological response and the other inhibits it. The autonomic nervous system has a central and peripheral parts. It is activated mainly by centers located in the spinal cord, brainstem, and hypothalamus. Also, portions of the cerebral cortex, especially of the limbic cortex, can transmit signals to the lower centers and in this way influence autonomic control. The centers of the sympathetic nervous system are represented by the lateral intermediate nuclei of the lateral horns of the spinal cord's gray matter from the 8th cervical to the 3rd lumbar segments. They are formed by multipolar neurons, the axons of which are part of the anterior roots. This is localization of centers of sympathetic nervous system. The centers of parasympathetic nervous system are located in two discrete distant parts of the central nervous system. Cranial department. The cranial outflow is made up of brain and innervate. Third nucleus or oculomotor causes constrictions of pupils. 7. Facial. Activate lacrimal glands, nasal mucous glands, salivary glands. 9. Glossopharyngeal. Activate parotid glands. And 10. Vagus. Regulate head, lungs, stomach, pancreas, small intestine, and the first half of large intestine. Sacral department. It is represented by the anupharynx nuclei of the from 2nd to 4th segments of the sacral spinal cord. The axon of its neurons form preganglionic nerve fibers that go to the peripheral autonomic ganglia and end in synapses on their neurocytes. The peripheral part of autonomic nervous system are formed by ganglia, nerve fibers, and plexuses. There are ganglia of first, second, and third order. The first order ganglia are called paravertebral. These are paired formations that run on both sides of the vertebral column and are connected to each other by intestinal branches. As a result, a paired sympathetic trunk is formed. Ganglia of the second order are called prevertebral. They are located along the course of the nervous plexus. These plexuses are located on the aorta and its branches in the neck as well as in the thoracic, abdominal, and pelvic cavities. They are formed by a combination of afferent as well as pre- and post-ganglionic sympathetic fibers. Ganglia of the first and second order belong to the sympathetic nervous system. First and second order ganglia belong to the sympathetic nervous system. The sympathetic ganglia, this is here a drawing of sympathetic ganglia, are covered with a capsule from loose connective tissue. They consist of multipolar neurons, different in size, whose dendrites are strongly branched. Axons form, this is ganglia, and axons form postganglionic nerve fibers without myelin. They are adrenergic, with the exception of nerve fibers that go to the sweat glands and some blood vessels that have cholinergic sympathetic innervation. Parasympathetic ganglia. This is sympathetic ganglia, innervate organs. Parasympathetic ganglia. Parasympathetic ganglia include the ganglia of the third order. These ganglia are usually located in the intramural nerve plexus or paraorganically near organ. In the wall of the hallowing organs, they lie in the submucous or in the muscle tunic between its layers. The main mass of ganglion neurocytes of the third order is made up of two types of Dogel cells. Here, Dogel cells. Dogel type 1 cell. This is a Dogel type 1 cell. Are motor neurons. Their axons form. The axon is long and form postganglionic myelin-free fiber that go to the innervated structure. They have a long axon, and so they are called long axons. Type 2, here type 2 Dogel cells are defined as aqua process cells. They have process of the same size, among which it is difficult to determine the axon. By functions, these are sensitive neurons. Their dendrites end in the innervated organ with a receptor, and the axon form a synapse with the dendrites of type 1 Dogel cells. These two types of Dogel cells form local reflex arc. Autonomic reflex arc. It begins with sensitive autonomic neurons located in the spinal ganglia. This is spinal ganglia, and here is a sensory neurosite. The second neuron of the arc is located either in the intermediate nucleus of the lateral horn of the spinal cord or in the autonomic nuclei of the brain. The neurons of these nuclei are called preganglionic. The axon leave the central nuclei and form preganglionic nerve fiber. These fibers go to the autonomic ganglia and form synapses on the efferent autonomic neurons. The axons of efferent neurons, this is axon of efferent neurons, form postganglionic nerve fibers that go to the innervated organ and end in synapses on its structural elements. Sympathetic reflex arc. Sympathetic reflex arc. It begins with a receptor that is formed by the peripheral process by dendrites of the pseudo-unipolar neuron of the spinal ganglion. Sensitive neurons of this reflex arc. Along this process, the excitation is transmitted to the body of the pseudo-unipolar neuron, and then along its axon, this is axon, to the multipolar neurons of the lateral intermediate nucleus of the lateral horns of the thoracolumbar segments of the spinal cord. The axons of these neurons, preganglionic, cholinergic nerve fibers, pass through the anterior roots of the spinal cord and go either to the paravertebral, such as in this drawing, or to the prevertebral ganglia, respectively ganglia of the first and second order. There they transmit excitation to the ganglion neurocytes. The transmission of arousal is blocked by drugs called ganglioblockers, which are widely used for the treatment of bronchial asthma, hypertension in the anesthesiology. The axons of ganglion neurons form postganglionic, myelin-free, adrenergic, sometimes cholinergic nerve fibers, which are usually very long. They go to the innervated organ, where they end in synapses on secretory cells or on smooth muscle cells. In this case, the excitation to the working structure is transmitted through the adrenoreceptor or noradrenoreceptors. Parasympathetic reflex arc. The sensory link in the parasympathetic reflex arc is more diverse. At first, pseudo-unipolar neurons of the spinal ganglia are sensitive neurons for the sacral part of the parasympathetic neuron system. Second, sensitive neurons of ganglia 3 7, 9, and 10 pairs of cranial nerves provide an efferent link to the cranial division of the parasympathetic nervous system, and Dogel type 2 cells are sensitive neurons of local reflex arc. Consider the reflex arc of the parasympathetic nervous system realized at the level of the sacral segments of the spinal cord. These reflex arcs begin with the receptor that are formed by the peripheral process of pseudo-unipolar neurons. Through this process, the excitation is transmitted to the body of pseudo-unipolar and the axon. Axon directed to the Onufrovich nucleus of the spinal cord. There they form synapses with the neurocytes of this nucleus. The axon of central neurons of the nuclei form preganglionic cholinergic nerve fibers that can pass through the para and prevertebral ganglia, but don't form synapses here, go through transit. They are directed to para and intra-organ ganglia, or the third ganglia, and form synapses on effectors neurons in organs. This is parasympathetic in organs. The axons of type 1 Dogel cells are very short, preganglionic. This is Dogel 1 type, and this is postganglionic nerve fibers, very short. And the N on the effector nerve ending on the smooth myocytes or glandular cells. Now, let's talk about difference between the sympathetic and parasympathetic parts of nervous system. Functional features of the sympathetic and parasympathetic parts of the autonomic nervous system are due to their morphological difference. The difference are as follows. At first, the centers of sympathetic nervous system are located locally in the lateral horns of the thoracolumbar spinal cord. Parasympathetic discretely in the cranial part of the brain and the lateral horns of the sacral spinal cord. Preganglionic fibers in parasympathetic nervous system are long and postganglionic are short. And in the sympathetic nervous system, as a rule, the opposite is true. Preganglionic nerve fibers in the sympathetic and parasympathetic nervous system are cholinergic, and postganglionic in the parasympathetic, cholinergic, in the sympathetic adrenergic. The substrate of innervation of the sympathetic nervous system is the entire body. In the parasympathetic, only the organs that carry out the intake and accumulation of energy. Therefore, the efferent part of the reflex arc of the sympathetic nervous system has visceral and somatic part, while the parasympathetic part has only the visceral part. Enteric, metasympathetic nervous system. Many internal organs, after cutting the sympathetic and parasympathetic pathway, and when after being removed from the body, continue to perform their function without visible changes. These organs include the gastrointestinal tract, airways, uterus, oviducts, heart, gallbladder, uterus. Functional autonomy is explained by the presence in the walls of these organs of ganglia of the third order. This is ganglions of third order. This is the basis for the allocation of metasympathetic system. The site of localization of the metasympathetic nervous system is the intramural ganglia line in the wall of this organ. Features of the metasympathetic nervous system. They innervates only the internal hollow organs that have their own motor activity. In the sphere of its innervation, there are smooth muscles, secretion epithelium, local blood flow, and local endocrine elements. It has a synaptic connection with the sympathetic and parasympathetic system and has no direct synaptic contacts with the somatic reflex arc. It has its own sensory link, type 2 Dogel 1 cells. This is sensory link, type 2 Dogel 2 cells. It isn't in antagonist relations with the other parts of the nervous system. It is more independent of the central nervous system that has the sympathetic and parasympathetic nervous system, and it has its own mediators, whereas neuropeptides and ATP. In contrast to the sympathetic and parasympathetic system, the enteric reflex arc more often consists of two neurons. First, afferent, which is type 2 Dogel, and second, effector motor cell, type 1 Dogel cells. The dendrites of Dogel 2 cells form receptors on the organ's wall, and the axons of the Dogel type 1 form a postganglionic nerve fibers and end with an effector nerve ending on the working structure. Difference in the structure of the autonomic and somatic nervous system. At first, the autonomic nervous system innervates the entire body. So it is characterized by ubiquitous distribution, whereas the animal nervous system innervates only skeletal muscles. The centers of the somatic nervous system, this is center of somatic nervous system, are represented by the motor nuclei on the anterior horn of the spinal cord, which lies segmentally throughout. There is a distinct segmentarity of the peripheral part. The nerves come out as a part of anterior roots of 31 pairs of the spinal nerves and 9 pairs of cranial nerves. Autonomic nervous system is characterized by the focal location of the centers in the brain and in spinal cord, the absence of strict segmentation of the structure of the peripheral department. The nerves come out as part of 4 pairs of cranial nerves, the anterior roots of the thoracic, 3 lumbar, and 3 sacral spinal nerves. Effectors, motor neurons, or in the animal autonomic nervous system are located outside motor nervous system, located outside the brain, the spinal cord, in the ganglia of first, second, and third order. The volitional impulse doesn't reach the working organ. In the somatic neuron system, these neurons lie in the central nervous system, in the anterior horns of the spinal cord, and the volitional impulse reaches the working organ. And the efferent pathway in somatic nervous system, it is only action of motor neurons. In autonomic nervous system, efferent pathway is binomial. It consists of preganglionic nerve fiber, axons of preganglionic neurocytes, and postganglionic axon nerve fiber that formed by ganglionic neuron. Thank you for your attention.